I really wish I wouldn't do that. We're going to be talking about how B cells find their antigen and interact with T cells to then differentiate and turn into different effector cells. So first we want to talk about how B cells even find their antigen. This starts in the secondary lymphatic tissue. We're going to have these things called follicular dendritic cells. And they will have antigen on them, just like this. And what happens is, if we're asking how antigen even gets to the follicular dendritic cell, it's usually brought by complement. So C3d will carry antigen, and it'll attach to the follicular dendritic cells. They have little, this is right here, this is a complement receptor, so they'll just bind to that, and then they'll have antigen all over this guy. So why is this relevant? B cells, so after development they'll have, this is a naive B cell. He'll be carrying his B cell receptor, so low affinity, be like IgM right here. It's low affinity, and these guys will find and bind to a specific antigen on the follicular dendritic cell. So it's just like one of the arms. Once it binds, it'll send a signal through these two guys, so IgM or IgA and IgB. Signal 1. After its initial signal, it'll find a T follicular helper cell. So T follicular helper cells are CD4 positive T helper effector cells. So usually after a T helper cell encounters its antigen, it'll differentiate into an effector cell, which is a T follicular helper cell. And these, in the name follicular, it's in the follicular region of the secondary lymphatic, which is good, so they can bind to the helper cells, or to bind to naive B cells. So they'll look like with their T cell receptor, and they'll have a CD40 ligand. Okay, let's go back to the B cell. So what happens after this? It'll take the immunoglobulin through endocytosis, split it up, and then present it via MHC2. So let me draw it. MHC2. And then the CD40 ligand will connect to the CD40 of the B cell, and that'll be sent the CD40 ligand through the CD40 will send signal to. There's other things like maybe the CD40, the CD4 that I could probably draw, but I mean, there's a bunch of things I can draw like the B7, but it's not pretty relevant. After this, binding signal 2 is sent, and then it will be turned into a centroblast. And these guys are in the germinal center of the secondary lymphatic. And there's just a bunch of them dividing. They'll do two things. It's important for somatic recombination, and they'll eliminate low affinity immunoglobulin. Eventually, they'll turn into centrocytes. Okay, also, this is the dark zone, germinal center, and this is light zone. These guys will test their brand new B cell receptors onto the second function of the follicular dendritic cells. They'll test their guys on this thing. So one of two things can happen when they're testing it. If it fails, apoptosis, so cell suicide. If it binds high affinity, it can redo this entire thing all again. These guys, it'll just keep cycling and cycling until it has such high affinity that eventually it'll become a plasma memory B cell. I also want to mention two things that can happen during this transaction between the T cell and the B cell. The T cell can actually influence the isotype of the B cell by sending cytokines. So maybe interleukin 10 can influence it to become a plasma cell, and interleukin 4 can maybe influence it to become a memory B cell. So, yeah.